Hello and welcome back to biology. So this week we will start on chapter 4, which is a tour of the cell. And we're going to go over this so many times, you're going to get so tired of it, but it's a really good practice because we'll be going over it today in the lecture, and then you'll also be covering pretty much the same content when you do complete your lab for this week. So let's go ahead and start. So this is the tour of the cell, and we're going to talk about body cells, so human body cells, and also the types of things that you will find in plants. So plants have cells as well. So we're going to point out some of the similarities and then differences between plant and human cells. So just a basic introduction of the cell. We'll talk about the nucleus and ribosomes. So the nucleus has to do with our stored genetic information, and that's the type of information that is passed on from parents to offspring. And then the ribosomes have more to do with protein production, which has to do with something called the rough ER, and then also kind of lipid production, which is fat production, which is the smooth ER. We'll talk about the different parts of the endomembrane system, energy converting organelles, and then the cytoskeleton and cell surfaces. All right, so because, of course, we're online, it's kind of hard to get the concept of a light microscope. I know you all did some of that previously in the lab before this, but what we use typically in a biological lab is something called a light microscope, and it can display living cells. So there's a way that we can actually take a slide of something, put it under the microscope, and actually see living cells moving. It's so interesting. There are also different types of microscopes. So there are ones that are more efficient, of course. They can see much smaller organisms, things like scanning microscopes, transmission electron microscopes, okay? We can talk about magnification, which is something that just increases the size of an object's image, so it's easier for us to actually see. And then resolution is more of the sharpness or clarity of an object. All right, so in the 1800s, early studies of cells led to something called the cell theory. So this states that all living things, I should say, are composed of cells. And then all cells come from other cells. Okay, so basic information. All right, so looking at just some of the comparisons between a light microscope, the unaided eye, and then the electron microscope. So unaided eye are things we can just see without any kind of magnification, just with your typical vision. So of course the height of a human, right? And then let's look at the light microscope. So a lot of the different types of organelles that we'll see within the cell today. And then electron microscope can actually see things that we really can't see, like viruses, protein producing ribosomes, and then some actual proteins. All right, so let's start talking about the cell. So the first part of the cell we'll talk about is kind of the barrier to the outside environment. This is called the plasma membrane. It is a phospholipid bilayer, which means it's composed of something called phospholipids. They have a phosphate head and then two little fatty acid tails, and it's something called a bilayer. So bi means two. So that means that we have two layers of these phospholipids that are composing that cell membrane. So we also have some proteins embedded. So these are more known as channels or carriers. They help larger objects actually get into and out of the cell. Other proteins can act as pumps using energy. So let's take a look at the plasma membrane. So you see right here we have our phospholipid bilayer. Right here at the top, these little gray circles, these will be the phosphate heads. And these little yellow tails you see coming down, those are the lipid or fatty acid tails. So the main difference between the two is that the heads or these phosphates are something called hydrophilic. Let me finish writing phosphates. Okay, these are something called hydrophilic. So hydro, the word hydro refers to water. Let me write that down for you. Hydro refers to water, and I'll put H2O. And philic refers to loving or liking to be around something. So if we call these phosphates hydrophilic, that means they love to be around water. So while the internal environment is a little bit different for these phospholipids, these fatty acid tails are actually something called hydrophobic. So remember, hydro means water and phobic means having a fear of something. So these fatty acid tails do not like to be around water. So that's why they're right here, contained towards the middle of this bilayer. So you see here the phosphate heads are exposed to water on the outside of the cell and the inside of the cell, while those little fatty acid tails are nice and protected on the inner part of that cell. And then here next, you can actually see the different types of proteins we might have embedded here in this plasma membrane. We have our channel protein, and this is the way that certain things can get into or out of the cell. And these proteins actually have hydrophilic and hydrophobic regions as well. So the parts of the protein that you see facing outwards, these are going to be the hydrophilic parts, the parts that like to be around water. And then any portion you see kind of embedded in here in the middle of those little fatty acid tails, those will be the hydrophobic regions of that protein. All right, so let's compare prokaryotic and eukaryotic cells. So let's go ahead and compare and contrast. So let's look at some of the similarities they have. So all cells have a plasma membrane, and that was that outer bilayer phospholipid membrane. They have DNA, which is that hereditary material. They have ribosomes that make proteins, and they have cytosol. Okay, so anytime you see this beginning of the word cyte, so C-Y-T, it refers to a cell. So cyte means cell, and then sol means more of a solution. So I like to think of it as more of a cell juice, and that's all the kind of liquid that you find within a cell, and then you have other organelles kind of embedded in it. So bacteria and archaea are something called prokaryotic, while all other life forms are something called eukaryotic. So prokaryotic, these lack a true nucleus, while eukaryotic cells have a true nucleus. Another difference between prokaryotic and eukaryotic cells is that prokaryotic cells are typically unicellular, which means they're composed of just one cell. Unicellular. And then typically eukaryotic cells are multicellular. Okay, so that means they're composed of many cells, and they're typically a lot more complex than any type of prokaryotic cell. Okay, so these eukaryotic cells, which means they have a true nucleus, which means that nucleus is actually enclosed in something called a membrane. So kind of think of little layers of things, a little sac protecting it. Okay, so like I said, prokaryotic cells are typically much more simple. All right, so let's look at a picture of a H. pylori. This is a type of bacteria that causes a lot of stomach ulcers and also GERD, which is gastroesophageal reflux disease. So maybe sometimes if you have a history of, let's say, heartburn, that's one of the symptoms of having H. pylori. So let's look at the different types of structures you will find for this prokaryotic bacteria. On the inside, you have a bacterial chromosome, which contains DNA, very, very tightly wound DNA. You have a nucleoid region, but it's not bound by a membrane. And that's why we say it lacks a true nucleus. You're going to have a plasma membrane, of course. You have your ribosomes, but there are a couple of extra things that you will not find in a eukaryotic cell. So for example, you do not have a cell wall in human cells. You do have them in plant cells, but not human cells. Human cells also lack a capsule. So think of a capsule of maybe a vitamin or medication you might take. The same concept here for this bacteria. All right, and then you might also see this word fimbriae. Okay, so referring to kind of little extensions coming from the cell. They help to sweep it along in a kind of a motion, a forward or backward motion. And then we have flagella. So both of these, fimbriae and flagella, they contribute to something called motility, which is a movement of the cell. So flagella help these little cells to actually swim along. All right, so eukaryotic cells are partitioned into functional compartments. So there are membrane enclosed organelles, and organelles means tiny organ. And these help to do all sorts of activities within the cell. So the nucleus and ribosomes carry out genetic control of the cell, while the other organelles, they're involved in the manufacture, distribution, and breakdown of molecules that include the endoplasmic reticulum, both the rough and smooth, the Golgi apparatus, lysosomes, vacuoles, and peroxisomes. All right, so let's go through a couple more things. And let me show you a picture. This is a picture of an animal cell. So in the middle, you have the nucleus, which is going to be the largest part or the largest organelle of the cell. You have the rough endoplasmic reticulum. You have the smooth endoplasmic reticulum directly attached to that. And you can't really see the difference ready apparently in here, but look for those little kind of pink ribosomes you see. That is indicative of the rough endoplasmic reticulum because it's studded with those ribosomes. You have the Golgi apparatus. Right here, you have your lysosomes. You have your mitochondria, your centrosomes with centrioles, the protective plasma membrane, something called a peroxisome, and then the cytoskeleton. Okay, so let me go ahead and compare that to the animal, we'll say the plant cell for this one. Okay, and I can tell it's a plant cell because it has something called a chloroplast. And chloroplasts are the main sites of photosynthesis. So let's look at some things in the plant cell that you would not find in the animal cell. So of course, chloroplast is number one because we as animals cannot convert sunshine to food, right? They also have a cell wall. You do not find that in animal cells. A plasmodesma. So this has to do with photosynthesis. So of course, we don't have that either. Okay. Something else, they have a large central vacuole, and this is mostly for storage of water, any kind of nourishment and or wastes. Okay, so there are three, we can say four main differences in this picture versus the human or animal cell picture. All right, so let's go over the nucleus and ribosomes. Remember, those help to carry out the hereditary information of the cell. So nucleus, remember, it's the cell's largest organelle and it contains the cell's genetic material. Okay, so that is in the form of something called DNA, and DNA directs protein synthesis via RNA. So how we start off, we have DNA. DNA is transcribed into RNA, and then this RNA is then translated into the language of proteins. Okay, so DNA to RNA to proteins. All right, another thing that you might find with the nucleus is something called the nucleolus, and this is the region of the nucleus that contains the RNA. Okay, take another look. So here is our nucleus, that large purple, typically more central structure. It's going to have that nucleolus, which has the RNA. It has a nuclear envelope surrounding the entire nucleus, more of a protective function. And this nuclear envelope has something called nuclear pores within it. And that helps for the passage of genetic material to enter and exit the cell. All right, next we have ribosomes. These are the sites for protein synthesis. So that means they make proteins. So they're composed of something called ribosomal RNA or rRNA plus proteins. So the way that proteins are actually made is, like I said before, they are translated from DNA to RNA to proteins. And proteins help to make up, they pretty much do everything within our bodies. Okay, so looking here, we have our ribosomes. So cells that have more ribosomes are typically going to make a lot more proteins. All right, so let's talk about the endomembrane system. So many organelles are connected to something called the endomembrane system. So these are the membranes that are within or inside a eukaryotic cell. And these different organelles are actually going to have their own membranes, okay? So they're kind of bound up in little sacs. All right, so a lot of these different organelles that are within that endomembrane system, they function in synthesis, which is more production of different things, distribution, storage, and then export. So let's go through a few of those. So first off, let's talk about the endoplasmic reticulum. All right, so this is directly continuous with the nucleus. So if you are looking for the ER within the cell, they're directly attached to the nucleus, which is going to be that largest organelle if you're ever trying to find them. All right, so the smooth ER, so called because it is not studded with ribosomes, it looks smooth. It synthesizes lipids and helps us or our cells deal with detoxification. Lipids, remember, those are fats, okay? So it helps to make fats. While the rough ER is more associated with ribosomes and protein production, okay? So you'll see those studded kind of all along, it's kind of folded up like this, and then you can see these little studs of ribosomes. So you know that is protein production, okay? All right, so next we have something called the Golgi apparatus. I like to think of this as the FedEx of the cell. It participates in modification, packaging, and shipping either throughout the cell or outside of the cell, okay? So think of FedEx, think of everything they do. They create a label, they stick the label on there. If there's something that's flammable, they take it out, they modify it, and then they package it up in a box and ship it out to where it needs to go. All right, so this is the FedEx of the cell. Okay, so the Golgi do look a little bit similar to the rough and smooth ER. The only difference is that it's not directly connected, as you see right here, the ER is. It's not directly connected to the nucleus. So that's a good way to tell the difference between the two. All right, so next we have lysosomes. I like to think of lysosomes as the Lysol of the cell. So imagine a can of Lysol and you have it and you're spraying everything down. It's there to kind of just eat up things that shouldn't be there, right? So different pathogens, microbes that could be there making us sick. So these lysosomes actually have enzymes that break down anything that's not supposed to be in our cells and also any kind of damaged organelle, okay? So they help to clean up the cell. So lysosomes are our Lysol, okay? So for example, here's your lysosome. Here's a vesicle, kind of just like a little bubble, and it contains some damaged mitochondria. This lysosome is going to engulf it, spit out these little enzymes, and then actually digest what should not be in there. All right, so vacuoles, these are just pretty much just large storage containers, okay? They can be contractile or they can be like in plant cells where you have that large central vacuole that stores tons of water, molecules, and waste, okay? So you do not have these in animal cells. Okay, so lastly, the last part of that endomembrane system is something called a peroxisome, and they actually produce something called hydrogen peroxide. So if anyone has ever heard of hydrogen peroxide before, or if you have some in your house right now, it's in those gigantic brown bottles, and you pour it on your skin when you have a cut or something, and it kind of bubbles up and tingles. So we buy it in the store, but our body actually makes it naturally. So it's something called H2O2 or hydrogen peroxide. And our body uses that to digest certain things and also to kind of further manipulate our lipids. All right, so that's the last part of the endomembrane system. So let's go over energy converting organelles. All right, so first let's talk about mitochondria. So these are different organelles within our cells that are able to make or harvest chemical energy from our food. So they actually carry out something called cellular respiration, which is the way that most eukaryotic cells make energy in the form of something called ATP, which is the same thing as adenosine triphosphate. So mitochondria have two internal compartments. There is an intermembrane space, and this space is between the inner and outer membranes, and I'll show you a picture in a second. And we also have the mitochondrial matrix, and this is going to have the mitochondrial DNA, which is separate from the cell's DNA. Okay, ribosomes, and many other enzymes. So the mitochondria is very self-sufficient. It has its own DNA, own protein-making machines, and its own enzymes. So let's take a look at that. Okay, so mitochondria looks like a little pill bug sitting right here within the cell. And you see we have this outer membrane, the intermembrane space, so that little space between the two membranes, and then the inner membrane. Okay, so within the inner membrane, we have that mitochondrial matrix and these little folds you see right here within that inner membrane. These are called cristae. All right, so another energy converting molecule that you do not find in animal cells are something called chloroplasts. So these are found typically in plants and algae. So photosynthesis is taking light energy from the sun and creating sugar and oxygen. So something called C6H12O6, which is a little simple sugar called glucose. And our animal bodies love to lose glucose to make energy. And then we also get our air, so oxygen, so O2 from plants, okay? So when we say that plants purify the air, they take the gas that we breathe out, which is called carbon dioxide, they convert it into pure oxygen. So that's why it's good to have lots of live plants around to help purify the air. All right, so there is a theory that we talk about with evolution is that mitochondria and chloroplasts evolved by something called endosymbiosis, which refers to this endosymbiotic theory that states that mitochondria and chloroplasts were formerly small prokaryotes that were kind of ingested into living larger cells. And they came with that theory because these both have their own DNA, their own protein, own enzymes, so are able to function independently. Okay, so just a little example of the ancestor of a eukaryotic cell. We have the endoplasmic reticulum, a nucleus. We're saying that this cell engulfed a mitochondria. Okay, and then gained the activity of that mitochondria. All right, so let's move on to the cytoskeleton and the cell surfaces. So the cytoskeleton is that it actually helps to organize the structure and activities of the cell. So remember, cyto refers to a cell, and then skeleton is the structure of something. So there are three types of little structures that are included within that cytoskeleton. There is the microfilament, the intermediate filament, and then the microtubules. So their functions include maintaining cell shape, anchorage, so being stuck in one place. So kind of think of an anchor on a boat. You kind of throw it out and it keeps you kind of steady in the water, right? The movement as well, amoeboid action, and then also muscle contraction. All right, so let's go over the microtubule, which is going to be the largest of the cytoskeletal components. Okay, here is a picture of an intermediate filament, which is more of an intermediate size. So in between a microtubule and a microfilament. Okay, and then last we have the microfilament, which is the smallest of those cytoskeletal components. Okay, another typical type of outer structure that you might see on a cell is something called cilia or flagella. All right, and these move when those microtubules bend. So both of these, the cilia and flagella, function in cell motility. So movement of the cell. Okay, so cilia are more hair-like. They're very, very short. Okay, they help to kind of just brush things along, while flagella are more of like a long tail that have a whip-like motion, and they help to steer and swim. The only type of flagella that you will find in human cells would be a sperm. So it has like a little tail, it has a little head, and then that long flagella tail to make sure that it can swim and also direct itself. Okay, so cilia are little hair-like movements. Okay, and then the flagella, which is this long piece that you'll find here on a sperm. All right, so here is the inner structure of a microtubule. You have just the way they're arranged on the outer segment, something called the outer microtubule doublet. You're going to have a central microtubule, some cross-linking proteins, some motor proteins called dyneins, and then also a plasma membrane. All right, so a couple more things dealing with the cell. The extracellular matrix of animal cells functions in support and regulations. So animal cells, they actually make and then kind of spit out something called an extracellular matrix. And this, of course, is on the outside of the cell to help it with its function, support, all that kind of stuff. It binds tissue cells together. It supports that plasma membrane, which is the outside of the cell, and it also helps with communication. Okay, so the ECM can also attach to other cells through membrane proteins called integrins. So let's go ahead and take a look. Okay, so you see everything out here. So right here we have our phospholipid bilayer, that is our plasma membrane. Inside the cell, you see we have our microfilaments of that cytoskeleton. And then outside of the cell, we have that extracellular fluid and all of these little support structures right here. Okay, so you're going to have things like collagen, a glycoprotein, which is a protein with a sugar bound to it, those integrins that help with kind of cell attachment. And then polysaccharides. So a polysaccharide means a long chain of sugars. Okay, so there are 3 types of cell junctions that are found within animal tissues. So adjacent cells are able to adhere, which means to kind of stick to each other, interact with each other, so pass information and molecules back and forth between them, and also communicate between specialized junctions that we have for these cells. So there's 1, 2, and 3. So first off, we have tight junctions, and these are binding cells together to form leak-proof sheets. So these are very, very tightly bound, and there's pretty much no holes or channels in between the two. Next, we have anchoring junctions, and kind of just like what they sound like, they help to anchor cells into the stronger tissues to help them from kind of breaking off or kind of being sheared off and damaged. Lastly, we have gap junctions. So these little junctions between cells that actually have little holes or channels. So they allow the free passage of ions, small molecules, and actually nervous impulses to jump quickly from cell to cell. All right, so looking at a picture, here's our tight junction. You kind of see this right here between these cells, very, very tight, forms it into a tight sheet. Anchoring junctions, giving that cell more structure. Okay, and then gap junctions, you kind of see right here, these little holes or pores, so that those molecules and nervous impulses can get through. All right, so let's talk about cell walls. So cell walls actually enclose and support plant cells. So of course, animal cells do not have cell walls, but plant cells do. And so these provide an extra amount of protection because it goes around the actual plasma membrane. So it provides that rigidity. These plant cell walls are composed of something which is called cellulose, and cellulose are really long chains of sugars, something known as glucose, that provides support for that cell. All right, so plant cells have little cell junctions called plasmodesmata, and these allow the different plant tissues to actually share water, any kind of nourishment, and also chemical messages, kind of like those gap junctions in the human cell. Okay, so these last few pages are just some of the more organelles that we have within the cell. And like I said, this is a good introduction. I also have that video posted for your lab work. Let me know if you have any more questions regarding this chapter. All right, thank you for watching. And don't forget to complete your Chapter 4 quiz.